So we're also going to talk about methane. Could I ask you please to raise your hands if you do not believe in global warming? Okay, so it looks like we all know that global warming is a serious problem. We all know it's very, very dangerous for our future. But when people think about global warming and climate change, they usually think about carbon dioxide or maybe water vapor. But did you know that the second most emitted greenhouse gas is methane? Did you know that methane's global warming potential is 25 to 32 times greater than carbon dioxide's on a 100-year time horizon? 36% of methane emission does not relate to any kind of human activities. These natural methane sources are usually wetlands, and most of them are undiscovered. I'd like to introduce you to Team Hanjani from Radnóti Miklós High School. We are developing a Methine Bio Sensor. We recently returned from Boston, where we were the only Hungarian team to compete in an international competition called iGEM. iGEM is a synthetic biology competition organized by MIT for mostly undergraduate students. More than 3,000 students from over 300 teams all around the world presented their projects ranging from cancer detection to solar cells. This competition has been held since 2004 and has grown from a few dozen of students to an event that fills a large conference center and is one of the most prestigious student competitions currently. This wasn't our first time. Our school had the chance to participate in 2014. Needless to say, this conference was an incredible experience for all of us, and we're very happy with the positive feedback of the judges. Despite the fact that we were among the youngest participants and had to compete with teams with much larger budgets, it was satisfying to see that our work was competitive even with international standards.
Okay, so I think for most research teams, topic choice is one of the most, if not the most difficult challenges during the story of their project. Well, this was the case with our team as well. We had some interesting ideas, such as engineering rubber digesting bacteria, or creating a bicycle lamp with fluorescent bacteria, or curing a silkworm disease with modified bacteria. But then, one day, on a team meeting, someone asked, "Where is this special fragrance coming from?" And this started a brainstorming around methane sensing, which, as it had turned out, is a key to the solution of some serious environmental and social-environmental issues. One of these problems is methane's greenhouse effect. It tends to be ignored that a huge amount of methane emission has natural origin, and as seen, most of the natural methane emissions are related to boreal wetlands, and most of them are yet undiscovered. Another problem is related to homemade biogas plants, which have increasing popularity among people in developing countries. They all carry the risk of methane leaking into the indoors if they are poorly insulated. Methane accumulation in closed sites can lead to explosions and, as a result, fatal accidents. Also, recently it has been proved that the methane content of exhaled human breath can be a good marker of wound healing after surgery. Hospitals in developing countries often face financial difficulties in purchasing devices designed for that purpose. So, what connects these problems is that methane sensing is crucial for their solution. Also, in case of all of the mentioned problems, the usage of existing electric gas sensors is limited by the lack of electricity. Therefore, our team has decided to create a methane biosensor.
For our purpose, we choose to work with methanotrough bacteria. This bacteria uses methane as its sole carbon and energy source. And amongst them, we choose Methylcoccus capsulatus, which is an obligate aerobe, thermophilic, gram-negative bacteria. We choose it because it's a well-researched bacteria. Our first plan was to insert a gene from Methylcoccus capsulatus into Escherichia coli. This gene is coding the enzyme methane monoxygenase, which is responsible for turning methane into methanol. Our plan was that after the insertion, in the presence of methane, we would detect the methanol accumulation with the help of an enzymatic assay. But we had some problems because MMO was too large and consists of multiple subparts, which would mean very low transformation and very low working efficiency for us in order to create the desired biosensor. Our second plan was to silence a gene inside Methylcoccus capsulatus. This gene codes the enzyme Methane Dehydrogenase, and it is responsible for turning methanol into formaldehyde. Thus, in the presence of methane, we would detect again the methanol accumulation. However, we had again multiple problems, such as we would cut Methylcoccus capsulatus from its sole carbon energy source. And also, we found out that methanol can be hardly differentiated from other kinds of alcohols with enzymatic assay. So we came to our final plan where we decided that we should measure the metabolic rate of Methylcoccus capsulatus. For this, we had to find an organic compound that is not used up by Methylcoccus capsulatus, but can be set as one of its products. We observed that lactate is not present inside Methylcoccus capsulatus's metabolic pathway, however pyruvate is, and we can turn pyruvate into lactate with the help of an LDH, lactate dehydrogenase. And at the end, we would detect the lactate accumulation in the presence of methane with the help of a lactate enzymatic assay.
Okay, but there are three important questions probably arising from you as audience. How did we know that there is no lactate in Methylcoccus capsulatus? How did we arrive to the final design of our plasmid? And how to transform this strange bacterium with this plasmid? The scientific journey of our team has been run around these questions, basically. Here, you can see the whole metabolic pathway of Methylcoccus capsulatus. Here is methane, and here is pyruvate. But you cannot see lactate anywhere. Therefore, it would be a great idea to put an enzyme into the bacterium which is responsible for converting pyruvate to lactate. This enzyme is called lactate dehydrogenase, LDH. To make sure that there is no LDH gene in Methylcoccus capsulatus, we searched for LDH gene parts in the Methylcoccus capsulatus fully sequenced genome. The result showed that the Methylcoccus capsulatus does not carry the LDH gene. Okay, LDH. But there are an enormous number of LDH genes and enzymes. Which would be the most appropriate for our purposes? Since Methylcoccus capsulatus is a thermophilic organism which lives optimally at 45 degrees Celsius, we thought it would be worth searching for an enzyme of a bacterium which lives at the same temperature. So we chose the LDH gene of Bacillus coagulans. This way, we could make sure that it would work as expected. Okay, now we had the gene. We just needed to switch it on. To enable the gene to function properly, we had to find a sequence which works as promoter in Methylcoccus capsulatus. A former participating team submitted a sequence which was described to contain a promoter sequence. We searched for this sequence in the Methylcoccus capsulatus fully sequenced genome. We found that there was a nearly 300 base pairs long region at both ends of which there were genes starting, and these nearly 300 base pairs long region must contain the promoter. To be able to carry out the scientific experiments, we needed the help of lots of people. For example, Dr. Laurie Giver, the vice president of Calista, showed us the site methanotroph.org, where we could find valuable information about methanotroph bacteria. Professor Gábor Rákely, head of the Department of Biotechnology of the University of Szeged, provided us the broad-host range vectors. We needed the help of Dr. Sergei Stoyar, who is a professor at University of Idaho, and he is an expert in transforming methanotroph bacteria. He sent us the detailed method of triparental mating. But what is triparental mating exactly? Triparental mating is a kind of bacterial conjugation. For triparental mating, you need a donor cell which carries the plasmid, and you need a helper cell which helps the donor cell to transfer the plasmid into the recipient cell, which is actually Methylcoccus capsulatus. To show you how funny science is, we would like to share this picture with you. In this picture, you can see that the triparental mating isn't unknown for mammals as well. Really, there are some whale species which need third parts help in their mating process. Okay, but coming back to serious science, Here you can see the broad host range vector PME gene 7, which we used in our study. It's a broad host range vector because it contains a promoter which can work in E. coli, and it can contain a promoter for Methylococcus capsulatus as well. On the right side, you can see our final plasmid construct. At both ends of the gene sequence, we put different restriction sites. This resulted a unidirectional gene sequence. We also did some same-sense base pair changes because one of the restriction sites could be found inside the gene as well. At both ends of the promoter, we put the same restriction sites, which resulted a bidirectional promoter sequence. It was needed because we did not know the exact orientation of the promoter. After carrying out many scientific experiments, we finally succeeded in assembling our final plasmid construct, which contained the vector, the gene, and the promoter. We have also transformed the donor cells, which carry the plasmid and will transfer it into Methylococcus capsulatus. During our laboratory work, we did many times GIL electrophoresis, mini-preps, GIL extractions, ligation, and PCR reactions, and we cultivated as many E. coli as we cannot measure. But the most interesting part of the lab work was the cultivation of Methylococcus bacteria. Therefore, we used a bottle which was filled with nitrile mineral salts medium for the methanotroph bacteria, and the methane supply was ensured by a balloon. So far, we can conclude that we managed to construct a plasmid in silico. We succeeded in creating the final plasmid, which contains the gene and the promoter. We have transformed the final plasmid construct into E. coli competent cells. We can cultivate Methylococcus capsulatus, and we have the exact plan of conjugation and methane concentration measurements. For measuring the lactate concentration, we ordered a colorimetric assay kit from Sigma Aldrich. Here you can see a sample diagram which shows not real data about the wanted measurement results. The enzymatic assay kit would indicate the enhance in lactate concentration.
For our team, safety and security measures had high priority. We always evaluated experiments and take consideration human health and environmental risks, and always took precautionary measures. During our project, we made the survey in order to better understand the public view of GMOs and methane. The survey was available online to fill out by anyone, and it was filled out approximately 250 times. It is notable that most of these responses come from Hungarian high school students. Some interesting results show that most people rather support GMO use in medicine and energy production than in food industry or chemical production. It was also interesting to know that most people assume methane-producing organisms are useful mostly for biogas production.
Our project was presented to our fellow students by displaying a poster summarizing it in our school for 2 days. It was mesmerizing to see how we awakened their interest towards synthetic biology. Additionally, 2 members of our team presented the project to the Department of Biotechnology at the University of Szeged. During our project work, we had the honor to present our research work to Nobel laureate Bert Zechman and Brain Prize awarded Arthur Connard. It was a great experience to talk about the project with highly recognized researchers. We also collaborated with other iGEM teams, as you can see, and 2 members of our team participated in an iGEM team meetup in the Netherlands. We would like to say special thanks for Gábor Rákhely, head of the Department of Biotechnology of the Hungarian Academy of Sciences. He showed us the way of plasmid designation and arranged us to be able to work with modular broad host range expression vectors, which were baselines in our project. Andrea Borbola, assistant manager of the school laboratory, also meant valuable help for us with her practiced advices, and we would like to thank also for her colleague Enikő Gulyás. We are grateful to Professor Lori Giver and Professor Sergei Stoyar for their priceless help in cultivation and transformation of the bacteria. Thank you also Biomatters Ltd for providing us with the molecular biology designing software Genius, which was a really grateful, helpful tool during our project. And we are really grateful to the Szeged Scientists Academy for ensuring us the possibility to take part in the iGEM 2017 competition.
Thank you for your time and attention. If you have any questions, we are happy to answer now.